have discussed the experimental proof of DNA replication, and today we discuss the mechanism of DNA replication. Okay, so you know, within human cell, the inside the nucleus, around 2 meters long DNA is present. 2.2 meters long. Okay, then when this DNA undergo replication, is it started from one end or then extending to the other end? You just imagine the 2.2 meters long DNA, its replication is starting from one end and proceeding to the other end. Okay, how long it will be, okay, and how troublesome it will be, that all things you will understand. Okay, anyway, keep in mind, the replication starts at different regions. Okay, these regions are specific, not only at one place it starts. Replication, DNA replication starts at specific regions known as origin of replication. Okay, in short, it is known as ORI. So what is the significance of ORI site? ORI sites are the sites where DNA replication begins. Okay, and this can be at many places in the 2.2 meters long DNA. And these locations are specific. Okay, it's not random here and there. These are specific locations in the DNA. Okay, then what happens at the ORI, origin of replication site? What happens at the ORI site? Okay, then just understand, this is a double-stranded DNA. Okay, and here is one ORI site, here is another ORI site. Okay, at the ORI site, at first the DNA uncoils. Okay, the two strands of DNA separates at the ORI site with the help of certain enzymes. We will discuss it. Okay, and it produces some bubbles. Okay, you can see some bubble-like structures. At the time of onset of replication, show number of bubbles on the DNA. That means these bubbles can be called as replication bubbles. Okay, where the replication begins is known as a replication bubble. You just see one such a replication bubble. Okay, that means the two strands uncoiled. That is why it created a bubble. Okay, and this type of a replication bubble you are going to draw in detail, and this bubble, this part only we take, one half. And if is DNA replication from this here to there, on the other strand it is from here to this word, upstream to downstream, here from downstream to upstream. If on one strand it starts like this and proceed. Okay, on the other strand it starts and proceed like this. That only we will discuss. And one half of the replication bubble we can call as replication fork. That is what we are going to draw next. One replication fork. That means this much region we are going to draw. Okay, shall we? Okay, so what is an ORI site? An ORI site is the region where DNA replication begins. Okay, if it is 2.2 meters long DNA, we have many ORI sites. Then at ORI site, DNA unwind to form bubbles or replication forks. So the replication fork we draw next. Okay, one strand of DNA, we are drawing it stretched like this. Then next strand of DNA. Okay, at the replication fork, what happens? Here all DNA is base paired, connected to opposite strand. Okay, then whereas this bases remain exposed, means single stranded. Okay, then this strand direction, I mean from the fork to upstream we tell the direction. This strand direction from this side to this side we tell, I mean from the upstream to downstream it is called. 3 prime to 5 prime direction. Opposite strand will be what? 5 prime to 3 prime direction. Okay, then listen. Okay, we are representing some enzymes here, and what enzyme is required to uncoil this? I mean, what enzyme is required to uncoil this strand to 2? It is known as DNA ligase or DNA gyrase. I mean, DNA helicase or DNA helical coil unturn. I mean, uncoiling. What is it? Helicase enzyme. DNA helicase enzyme is required. Okay. Then the DNA helicase enzyme action split the 2 strands, and the 2 strands are uncoiled and they remain separate. Okay. The first enzyme. There are some SSB protein, single stranded binding proteins also required to keep it separate. Okay. These are not allowed to again pair. They require some other additional proteins. We will discuss it later. Okay. Then understand which is the main enzyme involved in this process. The process fully is enzymatic. Which is the major enzyme required for the process is DNA polymerase enzyme. Okay. There are many types discovered, 1, 2, 3 and all. Mainly DNA polymerase 3 is the chief enzyme required for the polymerization. As the name suggests, it is polymerizing nucleotides to form DNA. In order to build the DNA, this is the chief enzyme. Okay, but understand the DNA polymerase enzyme. There are so many characteristics for this enzyme. First of all, this is a major enzyme. First of all, this enzyme acts very fast. Okay, if this enzyme acts very slow, we cannot even express our character. And this is an enzyme which is acting very fast. That means, in the case of the best studied one, for example, in Escherichia coli, it is adding about 2000 base pairs per second. Okay, I mean that much speed the processes. Within its lifetime, I mean it has to build a long DNA. In the case of E. coli, okay, about 2000 base pairs per second is the production of, I mean the activity of the DNA polymerase enzyme. That much fast it is. So, first point, DNA polymerase enzyme is an enzyme which is very fast, acting very fast. Second thing, it is the most accurate enzyme. Very important feature. This enzyme is very accurate. If it is not showing accuracy, if it shows a minor mistake, instead of one base, instead of adenine, if it is adding something else, it is leading to severe change. You have seen, okay, sickle cell anemia case. So that means this is the enzyme which should be functional very specifically in high accuracy. So this enzyme is showing high accuracy. So fast, accurate, and another important feature of the enzyme is this enzyme always acts in 5 prime to 3 prime direction. It can do the catalyzation. I mean, it can do the catalytic activity only in 5 prime to 3 prime direction. Okay, in the DNA strand, you know there is the sugar present. Okay, and the 3 prime OH is exposed. Third carbon OH is exposed. Only to the third carbon, the next, third carbon OH group, the next nucleotide can be attached. So the next nucleotide can be attached to its third carbon. Likewise, only to the free end, 3 prime end should be the free end. Towards that side only, the new nucleotides can be attached. So how we proceed? The polymerization proceed from 5 prime to 3 prime. The reason is always the 3 prime OH group is free for joining next, not the other side. So this side is free. 3 prime OH end is free. So this side catalyzation, catalytic activity will be there. So 5 prime to 3 prime it proceeds. From 5 prime to 3 prime the polymerization proceeds. So just keep in mind, this enzyme always act in 5' to 3' direction. Okay. Then another important thing, this enzyme requires high amount of energy to act. Energy expensive enzyme. Okay. How they get energy? The energy for the action is available as triphosphates. Okay. Nucleotide triphosphates. See, every nucleotides are available as triphosphates. That means here if is adenine, opposite thymine is required. Thymine is available as thymine triphosphate. But what we need is thymine monophosphate. Okay, when the two pyrophosphates are removed, the two terminal phosphates are removed, it gives energy for this activity. Just understand, the enzyme DNA polymerase is highly energy expensive, energy required enzyme, and the energy for its action is provided as nucleotide triphosphates. Every nucleotide triphosphate is an energy rich molecule. You know ATP, adenosine triphosphate, energy molecule. That molecule, if thymine is here, ATP comes. But ATP is not required. What we need is AMP only, adenosine monophosphate only. When it cleave and break and remove the two phosphates, that gives energy for the action. So the removal of pyrophosphate is giving energy for this enzyme action. The two terminal phosphate, pyrophosphate we call. Is it okay? How is energy satisfied for this action? Okay? Every nucleotide is available as triphosphate, and by the removal of the two terminal phosphates, energy can be used. Okay, so regarding the DNA polymerase enzyme, the main enzyme required for the action. We first discussed the one enzyme, DNA helicase, needed for uncoiling the two strands. Okay, the second enzyme is DNA polymerase enzyme. Regarding this enzyme, this is an enzyme which is showing high accuracy in action. Okay, not permitting any mutation, any scope for mutation, and it is an enzyme which is very fast. It can catalyze the polymerization of around 2000 base pairs per second in E. coli. That much fast it is. Within a second, 2000 base pairs are added. That much fast it is. Then this enzyme always acts in 5' to 3' direction. The reason also we discussed, on a 3' OH group there is free group which can create bond with the next. Then it is requiring high energy. That energy need is satisfied by making the reactants available as triphosphates. Okay, all triphosphates, when they remove the terminal two phosphates, release energy. Okay, then come to the process. The DNA polymerase enzyme cannot begin the polymerization. It cannot start. Polymerization means here the new strand should be built. The opposite strand should be built. In order to build the opposite strand, suppose if it's here adenine, if it's thymine here, guanine here. In order to build a new strand, what should be done? Opposite to adenine, thymine nucleotide should be brought. Opposite to thymine, adenine nucleotide should be brought. Then here, cytidine monophosphate should be brought. Then it should be joined. This is what is called polymerization. Building the new strand complementary to this strand. Existing strand bases are exposed. Okay, attaching the complementary bases or complementary nucleotides, new strand should be built. That is a process of polymerization. But the DNA polymerase enzyme cannot initiate, cannot start. It requires an RNA polymerase enzyme to start this. Okay, so the starter is what? An RNA polymerase enzyme. So listen carefully to the 3' end. Here this strand 3' to 5', and the new strand 5' to 3'. So the new synthesis is from 5' to 3'. So this is 5' end. Okay. At the 5' end, an RNA polymerase enzyme comes. What is this enzyme? Starting enzyme is RNA polymerase, and this enzyme produce a small stretch of RNA, not DNA. It can never build DNA. RNA polymerase can catalyze the bringing of RNA nucleotides only. So it built what? An RNA. The newly built RNA we can call as primer. Okay, RNA primer. See, when we do painting the wall, first we do what? Putty. Okay, the same thing. First a primer is stitched or joined, then further. Otherwise, when we do good cloths for stitching, directly we want to stitch a fresh cloth. We first try it with a old cloth if the machine is working properly. Similarly, here a small stretch of RNA is built first. Okay, so this RNA is known as primer. RNA primer. So this enzyme is also known as primase. What is the enzyme actually? This is RNA polymerase enzyme, also known as primase. Okay, if the RNA polymerase enzyme, when it is building a small stretch of RNA, then the chief can come. Who is the chief? Who is the chief here? DNA polymerase enzyme. The DNA polymerase enzyme comes. Okay, then this enzyme, primer is this one, okay, green color I have drawn it. This is the primer. Okay, then DNA polymerase enzyme comes. Are you getting this? This is the newly reached one is DNA polymerase enzyme. I will just represent here is DNA polymerase enzyme. Okay, the new one. I'll just give you an animation video also, then it will be more clear. So, first one, RNA polymerase enzyme or primase, it built a small stretch of RNA, it copied an RNA based on the DNA. Okay, then the DNA polymerase enzyme has come and it built DNA. DNA polymerase enzyme built DNA, not RNA. So, it built the opposite strand continuously. Okay, without any delay, it copy and it build the new strands based on this. Okay, it was adenine in the opposite strand, thymine nucleotide, like based on that complementary base pairing rule, it build the new strand. Here, the new strand synthesis occurs continuously, without any delay it occurs continuously. So, the synthesis is known as continuous synthesis. The strand is known as leading strand. Okay, that means the newly synthesized strand is known as leading strand. Are you okay? Once again, here in the strand with the polarity 3' to 5', the opposite strand synthesis occurs continuously. So the synthesis is known as continuous synthesis. Here, the DNA polymerase enzyme is the chief enzyme to do the polymerization, whereas it cannot begin the polymerization. So, the initial enzyme which attached to this point is an RNA polymerase enzyme, which is also known as primase. Okay, and this enzyme is not building what? DNA. Instead, it build or it polymerize an RNA, a small stretch of RNA. It is known as the primer, RNA primer. Likewise, whitewashing we are using primer. Okay, so this is the RNA primer. Then the next chief enzyme comes. That is the DNA polymerase enzyme comes. It builds the DNA. It polymerizes the addition of deoxyribonucleotides and build the new strand continuously. Okay, and this synthesis is leading to a leading strand which has no any break. But here in the beginning there is an RNA that should be removed. The primer removal is done by DNA polymerase 1, another type of DNA polymerase. Okay, primer removal is done by the enzyme DNA polymerase 1. All primers are removed by this. Okay, this should be removed. DNA should not have a piece of RNA with it. It should be removed. Then, I think this part is clear. Then the opposite strand. Scientists have found that the opposite strand synthesis is discontinuous. It is not occurring continuously. Instead, it is a discontinuous synthesis leading to a lagging strand. Here is formed a lagging strand. Okay, because the synthesis is discontinuous. It forms a lagging strand with so many pieces. That pieces are specially named based on a scientist. We will discuss it. Initially, here how the strand formation will be from 5 prime to 3 prime means from here to this side. New strands in this is not based on the template polarity. Here, this is the 5 prime end, towards the 3 prime end it is synthesized, the new strand. Okay, this is the 3 prime end. So, this is the 5 prime end. Here, this is the 3 prime end. Okay, here what happened? Initially, first RNA polymerase come. It produces a stretch of RNA. It stops. Then who will come? DNA polymerase will come. Okay. It produces DNA, but it stops. Okay. If it stopped, again next RNA polymerase is required. DNA polymerase cannot again begin. Okay. For beginning, this is required. It produces an RNA. It stops. Then DNA polymerase comes. It proceeds and it produces the DNA. Okay. Again it stops. Likewise, as fragments, DNA is formed. These fragments are known as Okazaki fragments. Okay. These fragments are known as Okazaki fragments, that is resulting in a lagging strand, not a leading strand, not a continuous strand. Here formed as many fragments. If DNA is formed as many fragments, there is no any use. It should be joined. Okay, that is must. The joining is done by, ligase enzyme, DNA ligase enzyme. Okay, here also the removal is done by, primer removal is done by DNA polymerase 1 everywhere. So this side is leading strand produced, opposite side is lagging strand produced. Okay, what is its reason is still uncertain. Anyway, understand the process of replication. This is the most repeated question. You have to understand the same way you have to draw the diagram for your exam. This is not exactly given in a textbook, but this way you have to present the diagram because all the contents are given in your textbook, but the diagram is not given clearly. In a different way, we can come to the concept and it will be more clear. So, DNA replication is considered as a fully enzymatic process. It is fully under the control of enzymes. Okay, it requires number of enzymes. There are mainly 4 major enzymes. Which are the 4 major enzymes? The first one, DNA helicase. Second one, DNA polymerase. Third one, RNA polymerase. Fourth one, DNA ligase. Is it okay? So this can be another way question may be asked. First enzyme, second enzyme, third enzyme, fourth enzyme. 4 major enzymes. Okay. The DNA replication will not begin randomly here and there. There are specific regions where the DNA replication begins. That region is known as origin of replication or ORI sites. Okay. At ORI sites, by the action of DNA helicase, the 2 strands of DNA separates. It results in a bubble or replication bubble or fork. Okay. Then, when the 2 strands uncoil and remain and it is a replication fork, we can start the action of the other enzymes. Okay. On the strand with polarity 3 prime to 5 prime, opposite strand synthesis occurs continuously without any break. It results in a leading strand. Okay. And the synthesis is called continuous synthesis. On the other strand, the synthesis is discontinuous synthesis. And here it forms number of fragments. DNA has number of fragments. These fragments are known as Okazaki fragments. Okay, then any case, the DNA polymerase is the main enzyme which can build the DNA. DNA polymerization can be catalyzed by DNA polymerase enzyme. So this is the chief enzyme. But it cannot start the process. It requires an RNA polymerase to start. This is also known as primase. It polymerize what? RNA formation, not DNA formation. So everywhere where the beginning is, there is stretch of RNA is formed. Then the DNA polymerase command proceed it. Then DNA will be formed. This is here only one RNA polymerase, a short stretch of primer, then full DNA. Whereas in the opposite strand, wherever there is break, the new action should be by RNA polymerase enzyme. So here RNA polymerase, RNA, then DNA polymerase, DNA formed, broken, then RNA polymerase, RNA, DNA polymerase, DNA, broken. Again, we have just shown a small area. Long area should be formed like this. Number of Okazaki fragments will be formed on. That all should be joined at the end by DNA ligase enzyme. Okay? And this is a very important process in our body. Any type of error will lead to a character disorder. So this is a highly accurate process. And if DNA synthesis doubling occur, but cell division is not coordinated, immediately there is no cell division. Cytokinesis means that is leading to another polyploidy problem. DNA doubling occurred, 2n became 4n or like, but it has not divided into two cells. Means one cell has become 4n, polyploidy. So everything should be done in a coordinated manner. Okay, so this is about the DNA replication, semi-conservative model, because one strand is all, only one strand is new, one strand is old, one strand is new, and this is also known as semi-discontinuous because only one strand is continuous, other strand is discontinuous. Okay, so see you in the next class.